Hi everyone, welcome to my channel Srinivasa Biology Tutorial. For this section to discuss NCERT line to line chapter Cell, the Unit of Life, part. The nucleus is a cell organelle, the first discovered scientist Robert Brown. The cell first to discover the scientist was Robert Brown is here in 1831. In 1831. The latter the material, the nucleus stained by as in the basic dyes was the given. The name is chromatin. The scientist was Fleming. The chromatin name they given by scientist Fleming. Okay. Now interface nucleus has been highly extended. The elaborated the nucleoprotein fibers is called as chromatin. So in interface, the total nucleus will be there elaborated, will develop like as in a complete the fibrous structures. It is called as what? Chromatin. The nuclear matrix and one or more the spherical bodies is called as a nuclei. So inside they have one or more spherical bodies also they are present. Those are called as what? Nuclei. Now here electron microscope has any relevant that the nuclear envelopes that which has consist of as a two the parallel membranes, the space between their 10 to 15 nanometers the perinuclear space. So they having as a two layers as present like here they having as any the two layers is present here like the two layers is present. In the two layers here in these two layers so these two layers is present like this space is called as a perinuclear space. It is called as what? Perinuclear space. From as in a barrier between as in a the material the presence the inside of the nucleus and that it is a cytoplasm. So inside they have is complete what? The cytoplasm is present. Now this basically the outer membrane usually the remaining the continuous with as a endoplasmic reticulums. Also it is a has in a ribosomes on it. The outer surface they having as what? Some in a that endoplasmic reticulums also present. In that endoplasmic reticulum also they consist of what? In a, the ribosomes as present. Now an amount the number of the place the nuclear envelopes are interrupted by as in a minute pores as present. So these minute pores also they are present. That minute pores are the in a passing through the which as a movement of the RNA and protein molecules takes place in both directions between nucleus and the cytoplasm. So molecules to move from as an inside to outside and outside to inside. The molecules to get movementation takes place cytoplasm to as in a nucleus. Normally in their one nucleus per cell, the variation of the number of nuclei are frequently to get observed. Can you collect when you recollect name of the organisms that we have more than one nucleus per the cell. So when that more than one nucleus is present in some organisms. Okay, now in some mature cells even lack of nucleus, example in erythrocytes. In many mammals like the sieve tube cells have in a the vascular plants which they having as a mini nucleus as present. Now the nuclear matrix of nucleoplasm contains as a nucleus and the chromatin is present. So inside of the nucleus they having as what? The nuclear proteins as present as well as in a the nucleus also they get present. Now the nucleus are the spherical structure present on the nucleoplasm. So they having as a spherical structure is present, that is called as what? Nucleoli. It is contained as in the nucleus, the content with as in the rest of the nucleoplasm, and it is also membrane bond structure, the site for as an active ribosomal RNA synthesis. So their major function is what? In a ribosomal synthesis. Ribosomal synthesis. Ribosomal synthesis. Now the large number more numerous nucleus are present on the cell actively carrying as a protein synthesis. When they are present, what happened? The cell actively they are carrying for out of their out protein synthesis. Okay. Now after you may recall that interface of nucleus, the loose, the distant, the network nucleoprotein, the fibers is called as a chromatin. So when we observe to as a here completely one chromatin is given. Here one chromatin is here clearly to get given here the chromatin, during the different stage of the cell division, the cells show as in different structures is called as in a chromosomes. They show as in a their different structure is close chromosome in that nucleus. Now, the chromosome contain the DNA in some basic proteins is called as a histones. So mostly they having as in that entire the DNA consists of as what? Some histone proteins as present. Non-histone protein parts also present in that RNA. RNA has in a non-histone proteins. A single human cell has an approximately the 2 meters of long thread-like DNA distributed as along the in a 46. There are 46 or there are 23 pairs of chromosomes as present. 23 chromosomes. 23 chromosomes means how many here? 46. There are 23 pairs means 46 chromosomes as present. Okay. Around as a 2.2 meters of the DNA is present. Now you study about detailed study of the DNA packing of in that form of in a chromosome in that class 12. Okay. Now they given as a one chromosome. Every chromosome visible. Let's see the under electron microscope. The primary constructed part is called as a what? Centromere. See in that entered the region. I am taking as a same this chromosome. In this chromosome. The central part is called as what? It is completely centromere. It is what? Centromere. And the side of which has the shape is structure. It is in a the side which has disc shaped structure is present. That is called as what? Kinetochore. So here one disc shaped structure is present. Here one disc shaped structure is present. That is called as what? Kinetochore. That is in a kinetochore. Now the centromere holds the two chromatids on the chromosome. They are based on the position of the centromere. The chromosomes can be classified to as in 4 types. So here the centromere, their position based on their chromosomes are basically 4 types. Okay, they are metacentric, submetacentric, acrocentric, telocentric. First to discuss metacentric chromosome. It is a middle centromere. The formation of 2 equal arms. So see here, they given clearly in this metacentric chromosome. Here, they are completely the centromere has in the middle and 2 arms are equal. And second one, the submetacentric chromosome has in the centromere slightly away from the middle of the chromosome, resulting one short arm and another is long arm. Here, in this process, one is in a long arm and another one is in a short arm will be taken form. That is in a submetacentric chromosome. Next, here in the case of acrocentric chromosome, the centromere is situated as in a close at the end of the formation of one extremely short and one is a very long arm. So one is a long, one is extremely short arm. That is an acrocentric. Whereas in a telocentric chromosome in terminal in the centromere has a terminal that is called as what? In a telocentric chromosomes. Okay, and in sometimes few chromosomes here some chromosomes has in a few chromosomes has in a non-staining secondary constructions also present. Here some chromosome has in a the secondary their constructed regions as present. This constructed regions present as in a terminal part. These are called as in a satellite chromosomes. These are called as in a satellite chromosomes. Like okay, these are the incompletely nucleus. The last one is microbodies. The last one is what? Microbodies. The many membrane bound minute vesicles are called as any microbodies. They contain as any various enzymes as present in the both plant cells as well as animal cells. So microbodies as mostly present in the plant cells as well as in animal cells. Okay. The remaining to discuss in next class. Thank you for watching.